Rainforests got their name for a good reason. Up to 200 inches of rain fall every year. That is 5 to 10 times more rain than in most temperate forests. People generally do not like getting rained on every day, but because tropical rainforests are hot, when in the field we often welcome rain to cool us. Rainforests are where most species of plants and animals live. These forests boil over with life, and most of the birds, mammals, and other smaller animals are active throughout the rain. Since 1952, Carl and Marion Rettenmeyer, assisted by many colleagues and friends, have had many opportunities to go to the tropics. Some people go to the beach. They go to tropical forests to study army ants, the most fascinating ants in the Western Hemisphere. The most aggressive of these army ants is the swarm raider Eciton burchellii. Anyone who first encounters the spectacular swarm raids of burchellii will be terrified and will never forget the sight of so many ants pouring across the ground. Here you see only a small area of about 1 foot wide. The whole swarm raid can be 50 feet across at the advancing front. Such a swarm may continue moving in one general direction for 6 hours. If you follow the ants backwards to find out where they are coming from, you may find their temporary nest called a bivouac. People sometimes think it contains millions of ants, but this dark mass actually has between 300,000 and 700,000 ants. It reeks like a wild hog with such a distinctive odor that bivouacs sometimes are discovered first by their smell. These army ants are unlike any other ants. Their queen never has wings, and she lays all the eggs for the colony in distinct batches called broods. Every 35 days, she becomes gravid or pregnant and lays up to 200,000 eggs in only 10 to 14 days. Then she takes a break and does not lay any eggs for 25 days. The larvae vary greatly in size, but are all about the same age. The smallest larvae become tiny workers, the biggest larvae become soldiers or majors. Every other size between those two extremes can be found in a colony. A colony kills and eats about 30,000 small animals every day. The colony eats so much that it would starve to death if it did not move every night when it has the immense brood of hungry, rapidly growing larvae. Nightly emigrations take the colony about 100 meters to new hunting grounds. The 14 days when the colony has a brood of larvae is called the nomadic phase. After the full-grown larvae spin cocoons, the colony stays in one location for 3 weeks. That period of 21 days is called the statelary phase. The army ants of fiction and Hollywood movies eat everything in their path: children, chickens, horses, entire villages. That is extreme exaggeration. However, Eciton burchellii does indeed eat thousands of different kinds of small animals, such as spiders, centipedes, and many kinds of ants. It kills an occasional frog, lizard, or small snake, but these are not eaten. It is surprising that such carnivorous ants have more species of small animals living in their colonies than are found in any other group of ants, wasps, bees, or termites. We have found over 200 species of insects and mites living with Eciton burchellii. These show many examples of how truth is stranger than fiction. We will show you a small sample of these species that rely on one species of army ants for their own survival. You might think that a bivouac of army ants would be ideal food for anteaters or other large mammals or birds. Anteaters feed exclusively on ants and termites. They eat them by the hundreds of thousands, but they prefer specific kinds of ants and termites. These anteaters obtain most of their food from ant and termite nests in trees. This tamandua anteater is an excellent climber and uses its prehensile tail like a fifth leg as it meanders through the trees. The rather fearless anteater will not even taste an army ant. The anteaters recognize army ants by the distinctive odors and leave them alone. This is the tongue from a tamandua that was found dead on a road. We placed a dead army ant soldier on the tongue to show you what could happen if an anteater made a mistake and tasted these ants. The giant jaws on the soldiers would stop the tasting instantly. The coati is another tropical mammal that eats many insects. Coatis will have nothing to do with army ants and flee the areas where they are raiding. Coatis try to escape from army ants and any other threat by retreating to trees. People who like to watch birds are attracted to army ant swarm raids because birds also watch these ants. Some species like this rufous motmot are frequent visitors. Motmots are famous for their distinctive tails. Near the end of the tail, some barbs are loosely attached and they are shed to leave the unusual tennis racket end. These motmots and over 100 other species of birds frequently visit army ant swarm raids. Although tropical forests have hundreds of species of birds that live primarily on insects, none is known to specialize on eating army ants. Among those birds, there are many called ant birds that forage for food by following the swarm raids of army ants. They are after the insects and other small animals that are fleeing the marauding ants. The great tinamou hunts for insects and seeds on the ground. It will pause briefly near the ant swarms. Tinamous are much more frequently heard than seen. They have one of the most beautiful calls, serenading us to sleep most evenings. The white-whiskered puffbird appears to have more feathers than it needs. Feathers cover its feet and are sometimes puffed up, making the small bird appear a little bigger. Puffbirds nest in cavities. They frequently excavate a nest cavity inside an arboreal termite nest. This puffbird gets its name from the sagging white feathers that look like a large drooping mustache. The white-whiskered puffbird is a frequent visitor to the swarm raids of Eciton burchellii. There are about 50 species of tropical wood creepers that are distinguished by their songs and the patterns on their breasts. This wood creeper has snatched a large centipede from the edge of the swarm raid. After quite a few hard whacks on the tree trunk, the bird swallows it whole. That seems to be a gigantic meal for a bird that size. Wood creepers forage with their breasts pressed against tree trunks. That does not help the birders trying to identify them. They have long tails with feathers that have sharp shafts that help the bird cling to tree trunks. 10 species of wood creepers regularly follow army ant swarm raids. One species, the white-chinned wood creeper, gets 95% of its food by following the army ants. Such birds have often been called professional antbirds, but the term obligatory is now preferred. According to Edwin Willis and his wife Yoshika Oniki, who have spent many years studying the behavior of antbirds, there are 46 species of birds that regularly follow the raiding ants. Many are so dependent upon the ants that they are at high risk of extinction if the ants become extinct. This male slaty ant shrike rapidly catches several moths or winged termites in quick succession. Those are two kinds of insects that Eciton burchellii does not eat. By flushing them out from under leaves or logs, the army ants help the antbirds. All these scenes of the male slaty ant shrike were filmed within 10 minutes while the bird followed an Eciton burchellii swarm raid. It is easy to see that the army ants improve the foraging success of the antbirds. The female slaty antshrike is less brightly colored than the male and often forages with her mate. The male comes back and shows common antbird behavior by tilting his head to look down at the ground. First he looks to one side, then to the other. The ocellated antbird has a bright blue patch around the eye. This individual is foraging at the ant swarm during a hard rain. If you watch closely, you will see the raindrops hitting among the raiding ants. All these antbirds spend most of their time standing around waiting for some arthropod to flee from the ants. They do not really hunt for food like most birds do that eat insects. We have never seen them fight with ants for the possession of a tasty prey. Occasionally, an antbird swallows an army ant that is holding tight to the prey as the bird swallows it. The bicolored antbird is a species that stays close to the ground during an ant swarm. We never saw it fly to a tree trunk, which is the standard behavior of the wood creepers. Its bicolored name comes from the contrast between its all-white breast and its dark brown back. The bicolored antbird is rarely seen away from army ant raids. Nesting success of this bird has been reported to be dependent upon finding a territory where burchellii raids frequently. The male spotted antbird is another species in which the sexes are different in color pattern. The male has a colorful brick red back with a striped tail. His breast has distinct black spots. This species has the frequent habit of jumping up a few centimeters, spinning 180 degrees, and landing right back where it started. The female spotted antbird is scarcely spotted. Pairs of spotted antbirds frequently forage close together at swarm raids. There are so many birds around a swarm front that birders frequently discover army ants when searching for the source of the bird calls. This male spotted antbird is about to defecate. It turns out that antbird droppings are an important part of the whole complex association of animals at army ant swarm raids. He appears to be checking out his latest contribution. Over 50 species of butterflies and skippers follow the army ant swarm raids. They come to feed on the bird droppings, which are an important, perhaps even essential, part of each butterfly's diet. You can look right through the wings on this glassy wing and see the leaf. That is a form of protective coloration that is provided by the lack of color because the butterfly's wings have few scales. There are many species of heliconia butterflies that have elongate wings. Some species are common around the army ant swarm raids. No one knows whether the butterflies find the army ants by the ants' odor or whether they are only attracted to the bird droppings. People who study butterflies know that some species of butterflies and skippers are more easily found around army ants than in any other microhabitat. This butterfly is in the family Satyridae, common butterflies found near the forest floor. Here a skipper is feeding on a bird dropping at a swarm raid. The skipper also defecates on the bird dropping. Flesh flies or Sarcophagidae sneak in and steal part of this tempting meal. Just as birders listen for the calls of antbirds, scientists who study flies follow the sounds of the flies to find the army ants. For every strange kind of animal on Earth, there is a scientist who studies it. Although ant flies are much more diverse and abundant than antbirds, people prefer bird watching to fly watching. There is little known about the flies, and there are no field guides for them. Tourists and scientists rave about finding 30 birds at an army ant swarm raid, but we have never heard anyone rave about the more than 1,000 flies at the same raid. There are over 60 species of ant flies, and practically nothing is known about their life cycles. They remain as poorly known as birds were 300 years ago. We have not taken the time to study hundreds of insects found with Eciton burchellii, but we did collect 22 species of flesh flies over burchellii swarm raids. We know that several species of these flies attack small animals killed and abandoned by the army ants. This one attacked an anolis lizard that the ants killed but could not eat. We reared 6 specimens of one sarcophagus fly from that small lizard. We reared a different species of flesh fly from a frog that the ants killed. We have no idea why the other flies are following army ants. Another ant fly is Stylogaster, a very elongate fly. The females have long pointed abdomens. Most of the time, they hover over the ant swarms. We have collected 12 species of Stylogaster over army ant raids. It is easy to identify the pointed eggs of Stylogaster. Here is an egg in the eye of a Calodexia, a common ant fly at the swarm raids. This Stylogaster egg is in a Calodexia abdomen. If you watch closely, you will see a Stylogaster hovering over the tarantula. People who study flies do not believe Stylogaster is a parasite of spiders. It is thought that they are parasites of other flies, but none has been reared. Calodexia are these pretty gray and silver flies with gigantic bright red eyes. They do not hover like Stylogaster, but instead perch at the edges of leaves, peering down at the swarming ants. They are very alert and have to fly or run to avoid the ants. We have never seen one caught by an army ant. When a katydid, cricket, cockroach, or large spider flees from the ants, the flies chase it. The fly eggs hatch within the female, and she drops microscopic larvae on the host, where they immediately bore inside and start feeding. There are about 20 species of Calodexia, and 12 of these have been collected only over swarm raids of Eciton burchellii. Often the most simple experiments are the most informative. We put thread leashes on katydids and cockroaches so we could reel them in after they fly from the swarm raids. It appears that the flies will not deposit their larvae on prey captured by the army ants, but only on the prey that is escaping. Our leashes make it possible to reel in the escapees. An attack by these flies takes only a fraction of a second, and the larvae are too small for us to see while we are watching a swarm raid. A few times we were lucky and caught a parasitized escapee from which we reared the adult flies. This is the only known attack of army ant flies on an escaping spider. You can see that the flies aggressively pursue the spider that tries unsuccessfully to kick them away. Unfortunately, the flies and spider all escape from us. Since the flies all appear to be the same size, they may all be one species of Calodexia. Near a bivouac, we find yet another group of flies. These are blowflies or Caliphoridae. They are probably attracted to the odor of the bivouac or the refuse deposit of the army ants. The bivouacs of burchellii have a strong odor that is distinct from the bivouacs of all other army ants. On several occasions, we found army ant colonies solely by smelling them. The refuse deposit or garbage dump is usually below the bivouac. The refuse deposits of Eciton burchellii also have a distinct odor different from that of the bivouac. If the bivouac is touching the ground, the refuse is carried about 30 centimeters or a foot away. The ants are neat and do not like living with their garbage. One can imagine that ant garbage may carry disease or bring predators to the ants. Empty cocoons of a brood of new adult army ants here cover the refuse deposit. When we see them falling, we know it is the day before a colony starts the next nomadic phase. When bivouacs are in trees, the ants sometimes carry the refuse down to the ground. Occasionally, this results in the ants doing an excessive amount of work. We have seen the ants carry refuse to the ground from a bivouac more than 30 meters up a tree. Army ant refuse deposits are teeming with life. You have to get down to ground level and explore with a magnifying glass to see the animals living there. An astonishing array of critters thrives on army ant garbage. One cupful of army ant refuse can include over 100 living animals. There is a lot of fresh meat in the refuse deposits because the army ants and their larvae cannot cut open hard body walls to eat the soft tissues inside. The ants remove these hard parts from the bivouac and put them in the refuse deposits. In this scene, the big obvious insects are the army ants. The workers carry their dead and deposit them in the refuse deposits without any burials. You must have sharp eyes and a magnifying glass to see the hundreds of beetles, flies, and mites. Most of them are less than 3 millimeters long and much smaller than the ants. The long, skinny beetles that race by are rove beetles or Staphylinidae. There are more than 50 different species that can be found in refuse deposits. Most of these beetles are not found in the emigration columns. We know that rove beetles fly into the refuse deposits after dark. They must find these refuse deposits by their distinctive odor. Even the experts who study Staphylinidae beetles cannot identify or don't have the time to name most of these beetles at this time. So these species remain nameless, like most of the beetle species on Earth. The rove beetles in refuse deposits have the appearance typical of thousands of beetle species found in leaf litter. However, we think many species are restricted to this specialized microhabitat. There are rove beetle larvae feeding in the refuse deposits. The mysterious large movements under the refuse are caused by fly or beetle larvae buried in the refuse. Rove beetle larvae are abundant, but have never been reared to adults. Therefore, we have no way to connect one of these larvae with an adult beetle, and hence no way to know what species it is. The most abundant flies are called scuttle flies or Phoridae. They are so small that we stuck one on a penny so you can see its size. When we zoom in from the whole penny to the date, you can see that adult. There, below the date, is an adult phorid fly. These scuttle flies are difficult to photograph alive because they are so small and you never know when they may stop running. And when they stop, it is only for a few seconds. When we zoom closer to the refuse deposit, you can see the scuttle flies zipping around in their characteristic zigzag run. These are all females with wings so reduced that they cannot fly. It appears that no phorid can run in a straight line more than about 5 millimeters. The same species of phorid flies found in the refuse deposits are found also running in the emigrations. While watching army ant emigration or raid columns, we discovered that many insects and mites run along in the ant columns or ride on the ants or the larvae or prey carried by the ants. Since the army ants are all aggressive carnivores eating thousands of different kinds of insects every day, it is strange to see so many different insects and mites living unharmed within their colonies. These insects have been called ant guests. even though there is no evidence that the ants invited them into their homes. By observing their behavior and how the army ants treat them, we can distinguish an ant guest from prey or insects we might pick up accidentally while collecting on the ground. One strange ant guest is a silverfish related to the ones that are common in the homes of people. This silverfish, Trichotillura manni, runs in the emigrations. It is known to live with several species of army ants throughout Central and South America. In laboratory nests, it runs all over the ants and their larvae. It feeds on microscopic scraps of food and whatever else is on the surfaces of ants. Since silverfish have a gradual metamorphosis, you will see small, medium, and large versions of the same species running around at the same time in a colony. By cleaning the ants, the silverfish may be somewhat helpful to the ants. However, the hundreds of thousands of ants in each colony are frequently cleaning themselves and each other. At the left side of the emigration column, you can see ants cleaning other ants. Cleaning seems to be a common behavior of the army ants both inside and outside their bivouac. They do more cleaning than eating or sleeping. In fact, we have never seen an army ant we thought was asleep. It is difficult to believe that silverfish can be much help to the army ants. As in the case with all the ant guests, the army ants provide all the food and protection for the guests. The ants sometimes attack the ant guests, but at all other times, the ants provide protection against all predators outside the colonies. Many rove beetles, or Staphylinidae, run along with the ants. Some look like thousands of species of rove beetles living in leaf litter, with no association with army ants. They show no obvious morphological modifications for living within an army ant colony, but all the species we have tested can follow the chemical trails of the ants. Some generalized rove beetles are predators on the adult workers. These rove beetles often run around with their abdomens curved forward over their bodies. They spray a defensive gas or microscopic drops of liquid from the tip of their abdomen. That gas appears to stun or irritate a worker and gives the beetle time to pull the worker away from an ant column. The beetles drag off dead ants, but are also predators on active workers. We think they are the most important predators on army ants. This Phyllanthus is the largest rove beetle we have found in any army ant immigration. But experts who study these beetles did not believe it could be an ant guest. Our photographs showing many of them running among the ants on immigrations convinced the expert that it is another undescribed new species of ant guest. The somewhat round beetles next to the large rove beetles are hister beetles. We will show you several of these large rove beetles as we see them in Eciton burchellii emigrations. They do not stop and pose. You have to be quick and don't blink or you will miss them. At least 4 Phyllanthus ran by in this quick series of video scenes. Did you see them all? It is difficult for the non-specialist to believe that this completely different looking beetle belongs to the same family Staphylinidae. Phthisis has a smooth, shiny dorsal surface that protects it from being bitten by army ants. The beetle can pull its legs and antennae under its protective shield, and we have seen the ant's jaws just slide off its back. Inside the bivouac, Phthisis feeds on prey collected by the army ants. Over 200 species of rove beetles are known to live within army ant colonies. This is the only one for which the larva is known. Phthisis larvae run in emigration columns and are most abundant at the start of the nomadic phase when the ant larvae are small. Although the beetle larvae do not look at all like the army ant larvae, we have not seen the army ants attack them. Probably they smell and taste like the army ants, and they are not recognized as an enemy. These larvae can also follow the ants' chemical trail, and you will see them run along after all the ants have emigrated. These are shown running at about 1/3 the actual speed. A third morphological group of road beetles has evolved to mimic army ant workers. Here we show you two road beetles that look more like the army ants than they look like their beetle relatives. To make it easier for you to see the beetles, we first show you each beetle motionless inside a circle. Then we show them running at about 1/3 natural speed. There are many different sizes, shapes, and colors of these ant mimics. Some are extremely good copies of ant shapes and have fooled many of our field assistants into thinking they were ants. In university classes in insect identification, these beetles are identified as ants. Some resemble their host ants in color, and others do not. In laboratory nests, we can watch these ant mimics climb all over the ants. The beetles constantly lick the ants, which might be somewhat beneficial for the ants. They occasionally rub their legs on the ants and themselves. We think this leg rubbing transfers army ant odor to the bodies of the beetles. Since army ants are blind, they must detect these invaders by touch, taste, or smell. We believe that natural selection by the ants has resulted in these ant-mimicking beetles. None of the ant mimics has been seen to attack army ants or their larvae. You may notice one pair mating as they climb around on the army ants. They feel right at home. While watching an army ant emigration, we see another group of tiny tear-shaped beetles running or riding on larvae or prey. These are featherwing beetles in the family Burchellii. The largest adult, Cephaloplectus, is only 3 millimeters long, slightly more than 1/10 of an inch. Here you can see one running in a lab nest. The scene shows us Cephaloplectus walking on a hister beetle. The Burchellii beetles all seem to feed by brushing microscopic particles off of adult or larval insects. These are the giants of the family, and the smallest featherwing beetles are only 0.8 of a millimeter long. That little running speck is a small Burchellii beetle. The running strides of one of these tiny beetles is about 1/100th of an inch. If the beetles run the entire emigration, that would be equivalent to a person running 3 Boston marathons every night, and they would have to do that every night for 2 weeks. Although we have seen these Burchellii beetles running in emigration columns, it is clearly to the beetle's advantage to hitch a ride. We first show you several Cephaloplectus running against the emigration traffic. As they do this, the beetles rear up and try to grab onto a passing ant. They often make many attempts before they succeed in getting a ride. Following those scenes, there are several riding beetles. Watch for the orange-colored spots riding on the ends of larvae or cocoons being carried. The Burchellii are especially attracted to the army ant queen. The last major group of beetles found with army ants are the hister beetles. Most of them are small, and some ride under the heads of worker ants. At first glance, it appears that this ant is attacking the beetle. It is actually giving it a bath by licking it all over. When the ant lets go, the beetle runs away unharmed and highly polished. We will show you 4 dead beetles at high magnification. These are between 1 to 3 millimeters long. The large hysterids are easy for us to tell apart in the field with a magnifying glass. We call them large because relative to the other beetles with army ants, they are large. In the entire world of beetles, they would be considered small, as they are only 4 millimeters long. We call this creature spiny butt, but its proper name is Symphilistra hamidi. The conspicuous hairy bumps on both sides of the thorax are handles by which the ants pick up and carry the beetles. The hind legs have large scattered hairs but no distinct orange brush on the inner surface. The ants rarely carry this beetle, but they spend much time licking them. We do not know whether the ants get any nutrition from licking any of their guests. Perhaps the beetles provide a drug that provides pleasure to an ant. We cannot tell if an ant gets intoxicated from licking any of the guests. The second largest sterid found with burchellii is Eciton. It also has handles on the thorax. However, the army ants rarely carry this beetle. The hind legs have a distinct brush of orange hairs. You will see this sterid brushing the ant's body with these hairs. This big beetle runs in the emigrations, but occasionally it hitches a ride on some big object the ants are carrying, like a major cocoon. This pair of Eciton was caught mating as they ran along in an emigration. This hesterid has interesting leg rubbing behavior. The hind legs of the hesterid are curved and have a distinct orange brush on the inner surfaces. With a microscope, we can see the ants are wiped with these brushes. A beetle will climb on top of a large worker or soldier. Then it rapidly alternates, rubbing its hind legs over the body of the ant and then over its own body. We believe the beetle is transferring the ant's odor to its own body. Here you can see Eciton using its hind legs to brush the head of a soldier. Relatively speaking, the beetles, silverfish, and flies I have shown you so far are the elephants of the ants' roving circus. We have been successful in getting only one photograph of the smallest insects that are guests of army ants. These are the springtails or collembola. But to my eye, it is the mites that are most interesting. They are the tiny clowns that ride on or are carried by the ants. They seem like clowns not because of their costumes, but because of their unbelievable shapes. They are the true morphological gems and are more numerous and diverse than any other creatures living with ants. In fact, we believe they are the strangest mites found on any animal. Here you see 3 pied-moted mites swollen with eggs into the shape of a small ant larva. All its relatives that are not found with army ants become gravid in the shape of a spherical balloon. The circle encloses the dark end. That is the size of this mite when it is not gravid. We have found tens of thousands of mites that tiny size. Among all the strange places where mites are found on army ants, one of the strangest is on the eye of a worker. Although we have found over 1,000 of these mites loose with ants preserved in alcohol, we never found this mite on an ant. Stephanie Berghoff and Eberhard Wurst took this picture of a live burchellii eye worker with this mite attached to its eye. The mite is so strange I predicted it would be found on some specific place. I never dreamed it would be found on an eyeball. This mite is Pinoglyphus, and it is such an unusual shape that it was placed in a new family, Pinoglyphidae. One family of mites, the Acidae, is ectoparasitic only on soldier army ants. These suck ant blood, similar to a tick on a dog. Ants have hard exoskeletons, and the mice have to find a membrane where they can insert their microscopic mouthparts. These mites suck blood at the base of the mandible of a soldier. We have never found them on smaller workers. They also feed on army ant larvae, but it does not appear to do them any damage. The biggest mites on ants are about equal to a rabbit-sized tick, on a German Shepherd dog. 5 new species of Eciton mites have been found on army ants since we began our work. Army ant mites are not only restricted to one species of ant, but have only been found on one specific area on one part of an ant's body. This mite, Eciton, has been found attached only to the antennae of burchellii. It is so elongate and narrow that the ants may mistake it for part of an antenna. Many other mites belong to a group of mite families called Euripidina. In 1926, a German mite specialist, Max Selnick, described 4 new species of Euripidine mites from Brazilian army ants. Their shapes were so strange that they have been placed in 4 different families. These mites have such strange shape and behavior that it is amazing that no one collected any more of them until we did extensive collections starting 30 years later. One genus, Burchellii, looks like a miniature half orange. When the mite walks, the tips of the legs just barely poke out beyond the back of the mite. The back is so highly arched that when you look at the mite from the side, you are really looking at its back. The bottom surface is concave and fits tightly against an ant larva or the body of a worker. Since the mouth parts and legs are completely hidden under the expanded dorsal surface, it is difficult to tell if the mite is feeding. Plantodiscus is another mite in this group Euripidina. Each of the 11 species of mites in this genus have a long ventral groove which fits against the undersurface of the tibia of a worker army ant. The head of the mite is always close to the tiny membrane between the femur and the tibia. That membrane allows the leg to bend at the knee, and the mites stick their mouth parts into that membrane. If you had a comparable parasite, it would be similar to a mite the size of a squirrel attached under your knee. We think an ant twitches its leg when the mite inserts its mouth parts, but once the mite is in place, the ants do not seem to notice it. A completely different mite, related to chiggers, we named the crimson giant. This is the largest mite we have found running in army ant columns, and it was in a column of Eciton burchellii. It is so brightly colored it is impossible to miss. Another outstanding mite discovery was made by Marion in 1956. While she was looking through about 10,000 live ant larvae, she said, "One of these larvae is walking." Of course, Carl said, "No ant larva has legs." She insisted, and what she was looking at has turned out to be a completely new family of animals previously unknown to scientists. This is the family Larvamimidae, named for the shape of the mites which mimic ant larvae. Even the hairs, which cover the body of the mite, are similar in size and shape to the hairs on the ant larvae. We have found 4 species of Larvamima, all with different species of army ants. This is the giant of the family. It is 2.5 millimeters, or 1/10 of an inch in length. Army ant workers treat these larviform mites the same as their own larvae, picking them up and carrying them on emigrations. Although these mites appear to be too heavy to move on their short legs, they are able to walk well. However, it would be impossible for these mites to walk 100 or more meters on the nightly emigrations, so it is essential that the ants carry them. Keep in mind, this living mite is only 1.5 millimeters long. All these previous mites have been found with Eciton burchellii. There are many more Euripidine mites found on other army ants. These include species found clinging so tightly to the basal segment of a worker's leg that they stay on the ant when they are preserved in alcohol. That leg segment is called the coxa, and the mites are called Coccyquosomidae. The back of this mite is so extremely expanded that the belly of the mite is sunk down in a groove. When you look at the mite from below, you see part of its back. We do not know where this mite rides, but its shape suggests that it probably was on a straight section of a leg. The genus Planodiscus now includes 11 species. They vary in shape, but all are easily recognized as belonging to this genus. The genus Antennaquisoma was named after an ant's antenna where some ride. A related mite, Antennaquisoma, has only been found on the outer surface of an army ant's tibia. We put dust all over the ant host so we could watch the ants clean it off. The ants cleaned down the leg and then continued to clean down the mite, apparently not noticing any difference. This mite has two posterior extensions that hold onto both sides of an ant's neck. The mite rides upside down under the ant's head, with the mite's mouthparts even with the feeding mouthparts of a large worker. We assume the mite samples whatever the ant is eating. One species of Cercospora has been found riding only on the jaws of the soldiers of a different army ant, Eciton. Not just any place on the jaws, but only on the inner curve of the jaws. It has a greatly concave ventral surface so that it somewhat wraps around the giant jaw. When the soldier bites, the long hairs on the back of the mite are touched, and the mite runs around and gets on the outer surface of the mandible. This is a bad place for the mite to ride during an emigration. If it is knocked off and lands on its back, the mite remains perched up on its long dorsal hairs. However, with its short legs, it is incapable of righting itself and will remain there until some ant knocks it over. Another large mite feeds by inserting its mouthparts in the small membrane between the tarsal claws at the ends of the hind legs of soldiers of Eciton. In discussing the extreme kinds of army ant mites, Hildobler and Wilson state that this mite, Macrocheles retinmyri, exhibits, quote, the most extraordinary adaptation of all the ant guests. The hind legs are held in a hook-like position. When the ant hooks its leg onto an object or another ant, the ant is really using the hook-like hind legs of the mite. Perhaps it is an advantage for the ant to be able to reach slightly farther, or more likely, the mite's leg provides a better grip. Insects are not only much bigger than the mites, but they are also much more rare in the colonies than the mites. We have found 327 species of insects, including 114 identified new species. We still have groups of springtails, beetles, and flies that have not been studied. We have studied these ants in only 5 localities. Since army ants range from the southern half of the United States to northern Argentina, there must be thousands of additional undiscovered species associated with these ants. Although we have found 167 new species of mites with army ants, this must be a very small fraction of the total species living with this small group of ants. We have collected over 75,000 specimens of mites from over 1,200 colonies of all species of army ants we have been lucky to find. This incredibly complex assemblage, ranging from birds to insects and mites, is the largest known association of animals on Earth. We now know over 200 species associated with Eciton burchellii. This association is the best example of how one species affects thousands of species in tropical forests. Even though Eciton burchellii may kill and eat 30,000 arthropods in a single day, it flushes out enough extra food to provide some food for over 100 species of birds, and 46 species are dependent upon that ant for more than half their food. As tropical forests are destroyed, army ants and thousands of interconnected species will become extinct. We hope this film will inspire more biologists, and especially children, to study ants and other arthropods. The land around every house on Earth is home to many creatures that have not been thoroughly studied. With careful observations and with good note-taking, anyone can add to our knowledge of our natural world.